Hello, everyone. Welcome to today's live broadcast, Biocompatibility Testing of Combination Devices, New Regulatory Guidance. This is Agnes Shanley, Senior Editor of Pharmaceutical Technology, and I'll be your moderator for today's event. We are pleased to bring you this educational web seminar presented by Pharmaceutical Technology and Biopharm International and sponsored by Eurofins Lancaster Labs and Eurofins Medical Device Testing. First, a word from our sponsor. With a proven track record of providing quality laboratory services for the largest medical device, pharmaceutical, and biopharmaceutical companies in the world, Eurofins Lancaster Laboratories and Eurofins Medical Device Testing deliver the industry's most comprehensive services to advance biopharmaceutical products, medical devices, and combination products to market. Now back to our program. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the left-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. It is now my great pleasure to introduce today's speakers. We are joined by Albrecht Poff, Jennifer Rourke, and Odette Mendez. With more than 25 years of experience in medical device testing and regulatory affairs, Dr. Albrecht Poff oversees laboratory operations, quality assurance, and regulatory procedures for Eurofins medical device testing laboratories across Europe. He currently serves as chairman of ISO TC 194 Technical Committee for Clinical and Biological Evaluation of Medical Devices and is head of the working group for ISO TC 194 Genotoxicity, Carcinogenicity, and Reproductive Toxicology. He also serves as chairman of the German National Committee DIN M12 for Clinical and Biological Evaluation of Medical Devices. Dr. Poff earned a PhD in toxicology and a master's degree in biology from the Technical University Darmstadt. Ms. Jennifer Rourke, Manager of Chemistry and Container Testing, oversees testing to support the container and package testing needs of both pharmaceutical and medical device clients. She has more than 21 years of testing experience and serves as one of the company's leading subject matter experts for extractables and leachables testing. She currently serves as the leader of two working groups for ASTM Subcommittee E55.04, leading the efforts to draft Standard WK 43945, Standard Practice for Determining and Characterizing Bioprocess Extractables from Components, Subassemblies, and Assemblies Used in Single-Use Applications. Dr. Odette Mendez is Director of Toxicology and Pathology for Product Safety Labs. She is responsible for direct technical oversight for toxicological and pathology assessment of preclinical testing. She provides regulatory guidance related to the pharmaceutical, medical devices, biotechnology, nutraceutical, and agrochemical industries. With more than 11 years of toxicology experience, Dr. Mendez is a diplomate of the American Board of Toxicology and of the American College of Veterinary Pathologists with extensive toxicology experience. Dr. Mendez earned a PhD in pathology at Texas A&M University and obtained her diploma of veterinary medicine at Lisbon Veterinary Medical School in Portugal. Dr. Poff, you have the floor.
Yeah, thank you very much, Agnes, for this kind introduction. Hello and welcome to Eurofins webinar on biocompatibility testing of combination products and medical devices. Our webinar will cover the following topics. First, myself will give an introduction on combination products, which will be followed by the risk management approach according to ISO 10993. The next topic will focus on the chemical characterization as an important step in the risk-based approach for medical devices. As a next topic, we will hear a presentation on biocompatibility testing, including some specialties for combination products. The webinar will end with concluding remarks and a future outlook. The topic of today's seminar is the biocompatibility of combination products which are combining a drug or biological with a medical device. The definition for drugs and medical devices are given in the respective guidelines in the EU and in the US, as mentioned in the slide. Combination products can be considered either as a drug or as a medical device. Combination products are defined in FDA 21st CFR 3.2 as products combining drugs, devices, and/or biological products together. The combination of a drug and a device or a biological is regarded as a combination product. Combination products also include co-packaged articles as a physically or chemically combined single entity. In 21st CFR 3.2, two rules are applying to combination products. The first rule, one application in case of a combination of two products. The second rule, primary mode of action of the product, as an important fact defining on whether it is regulated as a drug or a device. In this slide, several examples of combination products are listed. Combination product is a drug-eluting coronary stent, a prefilled drugs biologic delivery system, metered dose inhalers, a drug biological product packaged with a delivery device, but also a photosensitizing drug together with activated light. In the EU, an in-the-center consultation process is realized, and if necessary, an ombudsman will be consulted to decide how a combination product is going to be regulated. If the combination product is classified as a device, the notified body is the responsible authorized institution giving the CE mark, and the drug part is regulated by the European Medicines Agency. If the combination product is classified as a drug, either the national competent authority or the EMA will have the lead in this process, and the medical device part is evaluated by the notified body. In US, all combination products are centrally regulated from the Office of Combination Products. The combination products are classified by a formal request for notification, and the assignment is based on the primary mode of action, whereas the primary mode of action is defined as the single mode of action of a combination product that provides the most important therapeutic action. Certain challenges have to be considered for combination products. The safety and/or efficacy may influence or changed by the interaction of the drug and the device, chemically and physically. The device properties may change in contact to the drug by causing corrosion or by changing the mechanical properties. The bioavailability, stability, or purity of the drug may change in contact to or by application of the medical device. For new devices or for devices with material changes or modifications, the evaluation of safety will follow the ISO 10993 series. In case of already approved medical devices, the extent of the preclinical testing and evaluation will be focused on a potential new use of the medical device as part of the combination product. For the drug part of the combination product, the preclinical testing and safety evaluation is depending on whether the drug part is an already approved drug or a new molecular entity. If the drug is a new molecular entity, the safety evaluation is focused on the new molecular entity alone. If the combination product already contains an already approved drug for another use, the potential for a change in the safety, efficacy, and dosing requirements should be addressed in the evaluation. Important to consider is the potential interaction of the drug and the medical device. This is including the evaluation of the leachable and extractable profile of the device material into the drug. It's including any stability changes in the drug when delivered by the device or as a coating. It's including any drug adhesions or absorption to the device, and it is including also any potential breakdown products or manufacturing residues which may affect the safety. The focus of today's webinar is the biocompatibility testing of combination product with a special focus on the medical device part of the combination product. This slide is summarizing the important considerations for medical devices. As a key aspect is the evaluation of the leachable and extractable profile, but also the evaluation of breakdown products or manufacturing residue of the device material into the drug, which may influence the drug performance and safety. In addition, new devices or devices with material changes or modification, the evaluation will follow the ISO 10993 series according to the risk-based approach and or by testing. Extent of preclinical evaluation for already approved medical devices to be used in combination product will focus on the new use of the medical device as part in the combination product and will also follow the risk-based approach and eventually some testing. As already indicated in the next part of the webinar presentation, the risk management approach for medical devices according to ISO 10993 guidelines will be in focus. As already mentioned, the evaluation of the safety of medical devices is a risk-based approach. The application of risk management for medical devices is described in ISO 14971 as an overall standard. This standard determines a process on the evaluation of the hazards for medical devices, including the hazard identification, the risk evaluation, the risk assessment, the risk control, and the surveillance of the effectiveness of the control. Based on the publication of ISO 14971 in 2009, ISO 10993-1 was revised and republished in 2009. The title has changed from Evaluation and Testing to Evaluation and Testing within the risk management process. An annex, which is Annex B, was included in the revised version describing the identification of the biological hazards related to medical devices, their evaluation, their assessment, and the control of risks. In addition, also the supervision on the efficiency of the controls. The risk-based approach for medical devices is based on three very important standards of the ISO 10993 series. ISO 10993-1, describing the general principles of the biological evaluation of medical devices. ISO-18, providing information for the quantitative and qualitative characterization of medical devices. And ISO-17, giving guidance for determination of the allowable limits for leachable substances. The relevant topics for the biological evaluation are the materials used for processing of the devices, any additives, impurities and residues, any leachable components, any degradation product. But not only the chemical characteristics are important, also the physical properties of the medical device have to be considered, including porosity, particle size, form, surface morphology, all factors which can influence the biocompatibility of medical devices. The risk management process is including the risk evaluation of the biological or toxicological hazards based on Table A1 of ISO 10993-1. Here, the relevant toxicological endpoints are identified, which can be evaluated by literature, by clinical experience, or by testing. In the next step, the evaluation of the risk of the biological hazards will be evaluated. With this process, the hazards are identified and the risks of the hazards are estimated. A biocompatibility evaluation has to include a rationale for the performance of any testing or for a waiver of testing. A biological safety evaluation plan is required, which is considering all the identified hazards and their evaluation and the control of the risks in a clear, concise, logical, and scientific way. Everyone in the audience is aware of the table included in Annex of ISO 10993-1. In this table, the medical devices are categorized based on their application to the patient and their duration of use. This results in the selection of the toxicological endpoints which have to be considered for medical devices of different categories. From an historical point of view, this table was transferred from the normative main part of the standard to an informative annex. which was published in 2009 together with ISO 10993-1. This should indicate that testing for every X in the table is not really necessary. This slide shows the framework for the biological evaluation as included in ISO 10993-1. This starts with the chemical characterization and the chemical composition of the medical device. The evaluation can be carried out in 3 levels. The first level is the material toxicological equivalence of the medical device with an already approved product. The next level is the toxicological evaluation based on the chemical characterization of the medical device. And the third level is the biocompatibility evaluation, including the toxicological assessment and testing data. ISO 10993-18, the chemical characterization of medical devices, is described including a stepwise procedure in 5 steps. It is stipulated in Part 18 that analytical procedures have to be selected in order to get a robust assessment of the toxicological evaluation. It may be necessary that also new adequate analytical methods have to be developed, and if this is the case, these methods have to be validated, including precision, specificity, etc. This flowchart is included in Part 18, describing the 5-step process of the chemical characterization of medical devices. In the first step, the material will be qualitatively characterized, including the identification of materials and component chemicals, including residues or other relevant data. In step 2, the material equivalence of the new medical device to an already approved medical device is included in the risk assessment. If a material equivalence can be proved, the chemical characterization can be stopped. If a material equivalence cannot be proved, a full quantitative composition of the device is necessary as required in step 3. These established quantitative data will be used in step 4 to be included in the toxicological evaluation. This is based on a worst case assumption on the maximum concentration of chemicals in the composition if all is leaching out. If the worst case approach will not provide sufficient safety of the medical device, a use extraction to estimate clinical exposure can be carried out in step 5. These data again will then be used in the toxicological risk assessment. For the chemical characterization of medical devices, we have to distinguish between the characterization of the materials versus the characterization of extractable substances. The characterization of the materials is included in step 1 and step 3 of the flowchart, whereas in step 5, potential residual chemicals, extractables and leachables are detected and quantified. Within the last 10 years of applying Part 18, there was a development towards the extractable and leachables determination and an evaluation according to the PQRI approach for pharmaceutical closures. Another very important part of the ISO 10993 series is Part 17, which is describing a systematic process where the risk of hazardous chemicals in the medical device can be estimated quantitatively and the allowable limits of these chemicals can be identified. The evaluation is including the biological risk of leachable substances by collecting data and identifying critical health endpoints, by determining the tolerable intake levels specific for the route of entry and duration of exposure, and by also determining tolerable contact levels if irritation or sensitization is an endpoint. The toxicological evaluation is including the patient body mass and the application of utilization and safety factors. Also, the various routes of exposure have to be considered, including the uptake via the skin, oral or inhalative uptake, or the direct systemic application. Part 7 is also including a risk-benefit analysis, which can be applied if necessary. This slide shows the systematic process as outlined in the flowchart. It starts by identifying the leachable substance and the evaluation of the exposure, the duration of the exposure, and the uptake of leachable substances. In the next step, toxicity data will be collected and evaluated. In case adequate data will exist, the no observed effect level or lowest observed effect level can be identified for the key systemic endpoints. That for subacute, subchronic or chronic toxicity, and by selecting appropriate safety factors giving the tolerable intake for the non-cancer endpoints. In the next step, the tolerable contact levels can be evaluated in case the leachable is regarded to be an irritative substance. If the leachable substance is regarded to be carcinogenic, the tolerable intake can be also derived for such a chemical. In the last step, the tolerable exposure can be estimated by using the value for the tolerable intake, including the body mass and the uncertainty factors. In case the derived tolerable exposure is not feasible, a benefit factor can be included to derive allowable limit for the leachable substance. This slide summarizes the risk-based approach. The toxicological endpoints have to be identified according to ISO 10993-1 Table A1. The toxicological evaluation or risk assessment will be carried out according to Part 17 to derive the allowable limits for the leachable substances. But most important is the chemical characterization, which is the first step in the strategic approach for the biological evaluation highlighted in Part 1, but also the chemical characterization will provide information on the medical device, qualitative and quantitative composition, and the extractable and leachable profile, all included in the toxicological evaluation. In the next part of the webinar presentation, my colleague Jennifer Rourke will provide information on how to conduct a chemical characterization.
Thank you, Ali. My portion of the webinar presentation will focus primarily on chemical characterization of the materials used in medical devices and/or combination products. The key objectives of my presentation will be to first offer some background on the different classes of medical devices and to define the term combination product. I'll then discuss the importance of chemical characterization as one of the many phases of biocompatibility testing and highlight some key pitfalls to avoid. Next, I will illustrate the real-life challenges associated with chemical characterization through a case study example. Finally, I will relay current activities that are taking place and the direction in which biocompatibility testing will be taking in the future. Medical devices are divided into three classes. Class 1, which is low risk, minimum potential harm to the patient. Class 2, which are considered medium risk, require regulatory controls to ensure safety and effectiveness. And then Class 3, which are high risk, these must be approved by the agency to be marketed. They're typically implanted and used to support life. Our focus for biocompatibility testing and chemical characterization will be primarily for Class 2 and 3 medical devices, since they pose an elevated risk to the user or patient. And then there are combination products, which are defined as any combination of a drug product and a device, a biological product and a device, a drug product and a biological product, or a drug product, device, and a biological product. The challenge with combination products is the added complexity posed by the presence of both the device components as well as the drug product. Biocompatibility for combination products really spans both worlds, both biopharmaceuticals as well as medical devices. So why is chemical characterization important? The chemical profile can be used to assess toxicological risk. These data can then be used to evaluate long-term toxicity endpoints, for example, to determine if a chemical is carcinogenic, genotoxic, or results in reproductive toxicity. Chemical characterization can be used to identify intermediate and final breakdown products in a material, and comparisons can be made of the chemical toxicity profiles between a previously approved device and a new device. If the chemical profile is equivalent in a biocompatibility extract, then there may be no need to repeat testing using that solvent if the materials are the same. All tests have limitations. Some limitations of chemical characterization include the fact that surface properties of a device can affect biocompatibility. We need to know the identity of the chemical compound in order to assess its toxicity. Some minor peaks can be very challenging to identify, particularly if the mass spectrum is poor or the concentration is very low. And chemical characterization does not address the potential interactions between well-characterized base materials with polymers newly added to a device. However, the benefits can far outweigh the limitations. For example, we can use publicly available data to assess toxicology based on the threshold of toxicological concern per ICH M7 or ISO 10993 Part 17, based on the intended use or dose per day, as in the case of combination products. PQRI recommendations detail using a safety concern threshold of 0.15 micrograms per day for orally inhaled nasal drug products, or 1.5 micrograms per day for parenterals. Numerous guidance documents recommend just how chemical characterization can be performed to evaluate the biocompatibility of materials. These documents include the ISO 10993 Biological Evaluation of Medical Devices. Part 12 of that standard outlines how to perform the extraction of materials, talks about exhaustive extractions to ensure that no additives are missed. Then Part 12 discusses the various analytical techniques used to detect and identify these compounds in extracts. The newly released FDA guidance on the use of ISO 10993 Part 1 outlines the use of a risk-based approach to biocompatibility evaluation. The FDA guidance for container closure systems talks about how packaging for drug products must be evaluated for extractables and leachables. And finally, the USP has added General Information Chapters 1661 and 1663 to offer guidance on performing extractable studies on packaging. With all of these guidance documents, the central theme is risk. So what are the major considerations when designing a risk-based chemical characterization study? Let's look at the components associated with the device or combination product. Should the individual components of the device be characterized? This can be very helpful if there's a possibility of exchanging a component with an objectionable extractables profile for another. Or should the device be tested as a whole unit? This is helpful to evaluate only the drug contacting surfaces of a combination product. Let's also consider the solvents used for the study. What solvents are appropriate? Typical solvents include water, either acidified or alkaline water, a polar extract or a polar solvent such as isopropyl alcohol, and a nonpolar solvent such as hexane. What is the duration of contact? The longer the device is in contact with the body or a drug product, the more aggressive the extraction should be. And finally, what is the mechanism of migration? Is it from the device into the drug product or from the device directly into tissue, as would be the case with the Class 2 and 3 devices? The newly published FDA guidance on the use of ISO 10993 Part 1 requires that chemical characterization be performed on a device as a whole after the device has undergone all steps in the manufacturing process, including any washing or sterilization processes. So even if a study had been performed to evaluate the individual components of the device, an evaluation of the device as a whole must be performed as part of the biocompatibility study. Next, and this is very important, the solvents must not degrade or dissolve the materials. In other words, the solvents must be compatible with the components and only extract the additives, not break down the polymers. The extraction conditions should exaggerate and or simulate real-time conditions of use. Analysis of the extracts must include testing for the entire spectrum of organic compounds and inorganic or elemental impurities. And finally, is the method sensitive enough to detect the extracted compounds? The analytical evaluation threshold should be calculated based upon the safety concern threshold to ensure that the analytical method is sensitive enough to detect all compounds of concern. If not, then certain measures could be taken to concentrate the extract samples so that detection can be achieved. Next, I want to highlight a rather challenging chemical characterization study that we performed for a pre-filled syringe with a liquid drug product. First off, the syringe barrel, plunger, and tip cap were all polymeric materials. To further compound the situation, the active pharmaceutical ingredient was not soluble in water, so the matrix contained over 50% organic solvent. Therefore, the extraction was designed such that the syringe would be filled with 100% of the organic solvent, the same solvent that was used in the drug product formulation. Accelerated incubation conditions were used based upon the shelf life and actual use conditions of the drug product. The syringes were sterilized prior to testing, since sterilization is part of the manufacturing process. And finally, the extracts were screened for organic compounds using GCMS and LCMS, and also screened for elemental impurities using ICP-OES and an organic kit. The major chemical compounds observed in the extract were butyl oligomers, butylated hydroxytoluene, or BHT, palmitic and stearic acid, and an unknown polymer, which was later identified as a polyglycol ether detergent. The detergent was not so easily identified on the first pass analysis. The mass spectrum showed a polymeric pattern with a loss of 44 mass to charge ratio. Initially, we thought that the compound was something like polyethylene glycol. However, upon further investigation using accurate mass LCMS, we were able to determine that the likely empirical formula represented an alkyl ethoxylate compound. After some trial and error, using various reference materials for these types of compounds, the unknown was ultimately identified as a polyglycol ether detergent used by the manufacturer to wash the elastomeric stoppers associated with the syringes. This identification was confirmed using authentic reference material of the detergent, which turned out to be Genopol X80. In order to monitor the drug product for leachables from the syringe components, methods were then developed using LCMS to monitor for the phenolic antioxidant, the polyglycol ether detergent, and the fatty acid amides using commercially available reference standards. However, in the case of the butyl oligomers, no authentic reference material was commercially available. Therefore, a surrogate standard, tetradecane, was used for quantitation. This standard is structurally similar to butyl oligomers, so the relative response factor for the oligomers should be also similar to that of the tetradecane. The method validation was then performed using the spike recoveries of tetradecane in the drug product matrix to demonstrate accuracy. Moving on, I want to touch briefly upon some activities that are going on that will affect the future of biocompatibility testing. The USP Expert Panel on Biological Reactivity was formed in June of 2016 with the objective to revise the chapters based upon present-day technology and capabilities. USP would like to align with ISO 10993 with the goal of reducing the amount of redundant animal testing that is performed by first performing a thorough chemical characterization with evaluation of toxicity using publicly available databases and knowledge previously gained from toxicity studies. Where there is a knowledge gap, we would then perform cytotoxicity studies, and where there are failures for cytotoxicity or no previous toxicity data is available, we would then proceed with animal studies. The last slide of my presentation just highlights all of the general information chapters, guidance documents, and compendial testing chapters that are currently official or are currently being drafted, such as USP 661.4, which are plastic medical devices used to deliver or administer pharmaceutical products. I will now hand over the presentation to Odette Mendez, who will be presenting on biocompatibility testing.
Thank you, Jennifer. Good morning. In this part of the webinar, I will briefly describe some of the toxicology tests used for biocompatibility assessment. During medical device safety evaluation, all potential hazards should be considered based on the toxicology risk assessment, but it may not be necessary or required or practical to test for all these hazards. So the way that the testing is performed depends on whether there are short-term effects are needed to be evaluated for devices that have a short-term duration of exposure. And in that case, tests of cytotoxicity, acute toxicity, irritation, and sensitization may be required. And long-term specific toxic effects for devices that are going to be used for longer-term use, chronic and tests may be necessary, as well as implantation, genotoxicity, carcinogenicity, and possibly also effects even on reproduction and including teratogenicity. So this part of the presentation is going to focus on the tests that are common to all medical devices and that are the base for all the types and classes of medical devices that need to be tested, including cytotoxicity, irritation, and sensitization. Cytotoxicity is one of the main tests that is commonly used for biocompatibility assessment. Cytotoxicity is the capacity of a test substance or a test article or its extract to cause damage or death to a cell. The procedures for cytotoxicity for testing are in vitro procedures, and the test for the medical devices, depending on the type of medical device, may be a test that is performed with an extract of the medical device. But there's also some medical devices where direct contact tests or indirect contact tests be used and applicable. The objective is to evaluate whether there is an effect on the morphology of the cells from an intact monolayer cells to damaged monolayer cells. So the tests are usually done on extracts if the medical device usually is an object, and the medium and culture media is usually as the vehicle of extraction. This is then applied to the cell culture for a series of hours in time, and at the end of the exposure, there is an evaluation of the condition of the cultures and the percentage of the damage to the cell. Based on this, are deemed to be either cytotoxic or non-cytotoxic. So depending on the medical devices, and if it's possible, there's also the use directly of the medical device within the cell culture, either by direct contact when the material is placed directly in contact with the cells, and you can perform a qualitative or quantitative assessment of cytotoxicity, or an indirect contact, usually where the test article or the medical device is applied on top of a lean, say, agar layer, and this way you can also perform a qualitative assessment of damage to the cells. The next step of the testing for devices of short duration exposure would be irritation and sensitization tests. Usually all devices that will have patient contact will be required to have this type of testing. It is described under ISO 10993-10. The device and use of the medical device determines what are the required tests, and the reason these tests are used is because animal tests are good predictors of any potential issues that a device may cause. So starting with the irritation tests, irritation is usually an evaluation of a clinical sign at first contact. These effects are non-specific and inflammatory response, and depending on the usage of the medical device, you may need to have a single or continuous application of the material. There's different types of irritation. I'm going to go very briefly talking about each one of them, including skin irritation, intercutaneous, ocular, oral, vaginal, penile, and other types of irritation tests. For the skin irritation is for all devices that have a topical contact with the skin, where the medical device will be applied directly to the skin. This is usually performed in rabbits, and the scoring of irritation based on specific criteria of erythema and edema is done at different time points. Based on that, you're going to have a scoring of irritation that goes from negligible to severe. The intracutaneous reactivity test is just a little bit more involved, also using the rabbit, but here the medical device is applied directly inside the dermis, also score for edema erythema at the same time points, and usually the use of polar and non-polar extracts in accordance to the schematic design on this slide. Other tests of irritation that are common, if the medical device will contact the eye, there is usually performed an ocular irritation test, also performed in rabbits. And the evaluation can be done with one acute application or multiple applications, and the areas evaluated are the cornea, the iris, the conjunctiva. The results for this test are usually either positive or negative. Tests that are dependent on the local application of the medical device include oral irritation tests. If the medical device will contact the mucosa, and in this particular case, the Syrian hamster is the species of choice. And in addition to a microscopic evaluation for redness, etc., the irritation index is provided based on microscopic examination. That would be very similar to the vaginal and penile irritation tests that are performed in rabbits, and where, in addition to the microscopic examination, the microscopic scoring will provide the irritation index. Moving into the sensitization tests. These tests are designed to evaluate the potential for an immunological mediated cutaneous reaction of the test article. In humans, usually what we see is hitch, erythema, papules, and vesicles, or a combination of those. In animals, the erythema and edema are usually the main reactions that you see in the skin. There's three different types of tests. The guinea pig maximization test, or M&K test, that is widely used for medical device evaluation. The Buehler test, that also uses guinea pigs, and the local lymph node assay, or LLNA, that uses mouse. So the guinea pig maximization test is one of the most widely used tests because it is also the most complex. And the big difference is that there is an intradermal induction phase where the product or the test article is going to be applied directly into the dermis. 7 days after, you have a topical induction phase where you apply the test article on top of the skin topically, and then you have a challenge phase after 14 days. After that, you do the evaluation of the irritation score. And the guinea pig Buehler test is very similar, but there's no intradermal phase. There's only the induction phase. That means that you only apply the test article on top of the skin, and then you have the same challenge phase after 14 days, and you evaluate actually using the exact same scale for erythema and edema. The LLNA is a little bit different. It is designed under the guidance OECD 429, and it is an application on the ear of the mouse. And it's a dose-response assay, and there's a labeling with H3-timidine that gives you the possibility to actually have a quantification. It's called the scintillation or stimulation index, and based on this index, you evaluate whether or not your device is a sensitizer or an allergen. So briefly comparing this test, the maximization test is a test that is a little bit more involved. There's multiple steps, including intradermal application. The Buehler test is simpler, and the LLNA test is done in the mouse and gives you the possibility to have a quantification. If there is your contact of the device will be a long-term exposure, then it's very likely, based on your risk assessment, that you may need to perform systemic toxicity evaluation. That can be done on an acute basis. Administer the test substance usually less than 24 hours, or an extract of the test substance in case of the medical devices, or you can also perform subacute and subchronic toxicity evaluations. The chronic data, if chronic data is available, you may or may not do subchronic test. This really depends. If what your risk assessment is, what your materials are, what your chemical profile is, and based on this assessment, you're going to determine whether or not you need this chronic evaluation. The endpoints for systemic toxicity are a little bit more complex. In addition to usually body weights and clinical observations, there's usually a full systemic evaluation that is performed by doing clinical pathology, full clinical pathology panel, and full histopathology panel. And this provides you not just the point of contact, but any potential systemic effects of the medical device within the animal or with the extract application. If your medical device will be implanted, then it's very likely that implantation tests will be required. Implantation tests shall be used for the local pathological effects on the living tissue at both the gross and microscopic level, but also to evaluate systemic effects of implantation. And we need to characterize the history and evolution of the tissue response after implantation of the medical device or biomaterial, how it integrates, resorbs, and degrades into the body. So implantation tests, according to ISO 10993-6, will be designed based on the characteristics of the material. If the material is non-degradable or non-resorbable, usually a short-term response can be used 1 to 4 weeks, or a long-term response depending on the duration of the application of the material, up to 12 weeks. If it's a degradable material or resorbable material, depending on whether there's minimal or no degradation or when the degradation takes place, there is guidance on what the species should be used, and also what is the duration of the implantation exposure. There is also implantation guidance under USP, and usually the USP guidance evaluates the encapsulation of the medical device within the body. Usually it's either done in rabbits, where there's an intermuscular implantation, or done in rats for subcutaneous implantation, and the scoring that is performed microscopically is done depending on the capsule width. Another type of testing that can be performed for medical devices is when there is the question of concern, either by some of the extract components of carcinogenicity. In that particular case, genetic toxicology or genotoxicity may be warranted. Genotoxicity testing for medical devices is described under 10993-3. There are multiple tests that can be performed. One of the most common is the reverse mutation bacterialization assay, or AIMS assay, that evaluates for mutagenicity. The in vivo micronucleus test that evaluates for changes in the chromosomes or clustergenicity. And other tests commonly used are the in vitro mammalian gene test mutation assays using the thymidine kinase gene, or the in vitro mouse lymphoma assay, the genic and clustergenic endpoints. I'm going to talk to you today briefly about the AIMS assay, which is an in vitro assay that uses Salmonella or E. coli with or without metabolic activation that's used to mimetize the human. And basically, it's a rather simple test that will tell you if there is evidence of revertants that indicate a mutagenic effect. The micronucleus test can be performed in vivo and in vitro. The in vivo test, which is, I have a picture here of a cell, usually when is used to evaluate the alterations in the chromosomes, also called clustergenic effect. This is done in the peripheral, can also be done in the bone marrow, and basically the evaluation is the counting of these little dots that are there in the arrow that are a micronucleus. If this is a positive, or if you see an increase in compared to the control, then your portion of the extract or your material may cause alterations in the chromosomes, and further testing, such as carcinogenicity testing, may be warranted. My last slide is just to go briefly into just very brief discussion, because this has been touched, in terms of how do you test for a combination device. Combination devices are very complex, and there's a drug component. for which you will need to perform testing in accordance to the status of your regulatory submission. If you already have a drug that is previously approved, there may be need for additional bridging testing or may not. If you do not have, then the full battery needed by the regulatory authorities for the drug portion may be warranted. In terms of the device component, and this is what I've been talking mostly, it is a risk-based approach based on what my colleague Ali and Jennifer has said. There, you will design on your risk assessment strategy what are the tests that are needed, and they can be as simple cytotoxicity or more complex depending on the risk assessment. There's also container testing that Jennifer touched on, and that is based on both the regional pharmacopoeia and USP and also FDA guidance. This is all I have, and I would like to now pass this to my colleague Ali Poth for the conclusion and future considerations. Thank you.
Yeah, thank you very much, Odette. In the last part of the webinar presentation, I will provide some concluding remarks, and I will provide also a future outlook on the risk-based approach. And the three presentations have shown the risk-based approach for the biological and toxicological safety of medical devices. This approach has to include the identification of the relevant toxicological endpoints according to ISO 10993-1, Table A1. We have seen that the chemical characterization is the major element within ISO 10993-1, which will provide information on the qualitative and quantitative composition of the medical device, but also on the extraction and leachable profile. We saw that Part 18 is a very essential part. Based on the results of the chemical characterization, ISO 10993-17 is used to derive the allowable limits of any chemical of concern. Biocompatibility testing is also necessary for filling data gaps and for enhancing the safety of any medical device. The three major parts, 1, 17, and 18, are going to be revised, and I will highlight the most important changes in these standards in the next couple of slides. These changes will have an impact for the medical device industry and will bring new challenges in the future. Part 1 is currently under revision. The revision is based on the revisions of Part 17 and 18 at the same time, but also based on the publication of the US FDA document in June 2016. The important main change in Part 1 is the proposal of changing Table A1. This is the proposal for the new Table A1. Additional columns are included which follow the Table A and B included in the US FDA document. As an additional column, the physical and/or chemical characterization is included. Several additional toxicological endpoints are added for certain type of devices following Table A and B of the US FDA document. The table is indicating an X for the physical and chemical characterization of the medical devices, and other toxicological endpoints are registered with an E. The meaning is that first of all, a chemical characterization has to be carried out, and based on the results and outcome of this characterization, the toxicological endpoints related to the type of devices are evaluated on a case-by-case basis. This is again indicating the unique role of the chemical characterization also in the future in the biological and toxicological evaluation of medical devices. As already mentioned, Part 18 is going to be revised as well. Within the 10 years of experience with the standard, strategic aspects have been developed, including relevant, complete, and strict characterization for extractable and leachables of medical devices. Also, new concepts in risk assessment, for example, threshold of toxicological concern concept, as we already heard, are discussed and will be included in Part 17, which may also impact Part 18. Part 18 has to be seen in context to Part 17, as Part 18 is delivering the data for the toxicological evaluation. A more detailed description of the extraction procedures for exhaustive, exaggerated, and simulated use extraction will be included in the revision of Part 18. The standard will be revised completely. Definitions will be included for product configuration, and material composition. As already mentioned, the extraction methods for the evaluation of extractable and leachable substances will be described in more detail. The revision will also include a more detailed description of the stepwise characterization process and the respective changes in the flowchart. The analytical evaluation threshold, as we already heard, will also be included. Below this level, a chemical is exempt from the toxicological evaluation, where this level can be based on the TTC concept, for example. This slide shows the PQRI and the ISO approach together. The ISO approach foresees an extractable and leachable study in parallel in a qualitative or semi-quantitative way. The result of this approach will be included in the risk assessment. After that step, there is a question mark, which is discussed now within the expert group of ISO TC 194, the committee dealing with the ISO 10993 series. The further approach can include a quantitative assessment. It can also include relevant matrices, for example, sweat, blood, saliva, but also the determination of release kinetics, which can help to carry out a more reliable and more robust risk assessment. Also, Part 17 will be revised. For this standard, significant changes are needed to include new concepts of risk assessment. The instruction to establish a safe human dose for chemicals detected in the chemical characterization will be included. Also, an instruction describing the process for determining a margin of safety between patient exposure and tolerable intake is discussed to be included in the revision. A new concept for risk assessment is the threshold of toxicological concern for medical devices. This threshold value is valid for a group of chemicals which then should no appreciable risk to humans. This concept was developed to define an acceptable intake for any unstudied chemicals that will not pose a risk of carcinogenicity and other toxic effects. The TTC is then de facto regarded as a tolerable exposure value. The TTC is defined to 1.5 microgram per day intake of a genotoxic impurity in case of lifetime treatment. Some structural groups, aflatoxin-like azoxy or nitroso compounds, were identified to have such a potency below the TTC would theoretically be related with a potential risk for cancer. There are still some issues and concerns about the TTC concept. There is still a discourse on unknown substances found in the extractable and leachable studies, as the TTC concept is not applicable for some substance classes, and it cannot be excluded that some unknown substances may also be included in these classes. It has also further discussed if the concentration of the chemicals should be provided as a daily release or as a total amount of the substance in the medical device. The use of the TTC approach is still open. Should it be used as a screening method or should it be used as a standard TI value? The discussions are still open on the validity of the TTC concept, also for non-oral applications, as the TTC concept was developed based on oral applications. The planned changes in the revision of Part 17 will include a new work item proposal for the inclusions of the TTC concept in an annex or in a separate document. The TTC concept is also important for deriving analytical thresholds. Below that, no further toxicological characterization is necessary for an unknown substance. Within the working group, it is also discussed to include TTC values for other toxicological endpoints, for example, sensitization and irritation. The impact of the revision of the main standards for the manufacturers are listed in this slide. There will be a cost and time reduction for avoiding subacute, subchronic, or chronic toxicity studies by including the TTC concept in the toxicological evaluation. There will be an increased use of chemical analytical methods to identify extractables and leachables, which may lead to a cost reduction in case particular toxicological endpoints can be evaluated without testing. However, it can also lead to higher costs in case an extended chemical analysis has to be carried out for the identification of certain chemicals. Based on the US FDA document, additional endpoints have to be evaluated based on chemical characterization data, which may result in increased costs for the toxicological risk assessment. At the last point, which is very important, is the acceptance of this approach. We have to secure that this approach will have global acceptance, including not only Europe and US, but also China, Japan, Korea, and others. Yes, thank you for your attention. And with this, I will give back to Agnes for concluding. Thank you.
Yes. Thank you so much, Ali, and thank all of you for those outstanding presentations. It's now time for audience Q&A. Before we get started, just a very quick reminder to please submit questions by typing them in the Q&A box on the right-hand side of the window. Please know we have a limited amount of time. If we can't get to all of your questions, our expert speakers have agreed to respond to them individually offline. First question is for Odette. If I need to perform genetic toxicology testing, which test should I perform? Hello?
Yes, I'm sorry. Yes, it can definitely answer that or try to answer that. So if there is a medical device that is a combination of a drug product and there has a medical portion, then the genetic toxicology testing for the drug will be as the requirements of the agency to which has been submitted, or if the drug has been approved, then that we assume that is not further necessity. But for the medical device testing, it will be based on the risk assessment and on the characteristics of the extracts and the chemical characterization to see if there's any potential genotoxic materials in that list that have not been previously classified, or a TTC cannot be approach cannot be used. For the medical device itself, multiple tests can be performed. One of the most commonly used is the Ames test, and that can be followed either by the TK assay or the in vivo micronucleus, depending on the regulatory agency to which the submission is going to occur.
Great, thank you. Another viewer asks, can you please expand on irritation testing with respect to nasal irritation mentioned on the slide?
Sure. So all the irritation tests are based on the same principles. If the nasal cavity is where the device is going to be implanted, then the testing is going to be performed within the nasal mucosa. Application can be performed depending on the intended clinical application as a sole application or a repeated dose application, and the evaluation will be done not only on macroscopic evaluation, but the final scoring will be performed assessment and scoring of the irritation index.
Great, thank you. A question for Jennifer. During the extraction, the polymeric material visibly breaks down or swells. Can the extract still be tested?
Yes, hi. So if that observation is made, if we see that the polymer starts to break down, there's lots of flocculant in the extract, or conversely, if the polymer absorbs the extraction solvent, typically that's a sign that the solvent was not compatible with the material. And let me reiterate that it is very important that we're not breaking down the material. We just want to accelerate or exaggerate the conditions so that we can pull out additives. We don't necessarily want to just completely dissolve the material in the extraction solvent. So if that is observed, typically we recommend to abort the test and not to go ahead and screen the extract using the various techniques to screen for organics or inorganics. And typically we would then just note that in the final study report. However, if you really want to continue with testing, we can do so. The problem with that is it's going to be quite difficult to identify the additives at that point. The chromatogram might be quite busy, and meaningful information is likely not to be obtained. So we really recommend that extract be aborted at that point. If the polymer swells and absorbs the extraction solvent, sometimes they swell so much that you can't get any viable solvent out of the test. In that case, again, we would note that that observation was made and abort the testing. Or if you can get enough extract out of the material to proceed with the test, you could possibly do that. But again, beware that the results may not be meaningful.
Thank you. We have time for one more question. Ali, this is for you. What is the timeline for the revision of the 3 major standards?
Yes, the timeline is a little bit different depending on the parts because part 1 is going to be published at the end of 2017. Because not they are working a lot, but they are forcing this because it's an important standard. And the other parts, they will be a little bit delayed. It was an idea of some national mirror committees to have the part 1, part 17, and part 18 published simultaneously. As I think I have provided this, as these 3 standards are very well interconnected, and but this was not accepted by the majority of nationalities, and therefore part 18 and part 17 there will be the revision will be available. Maybe 2018, and part 17 maybe in 2019. This is the current information I can give.
All right. Well, thank you so much. Unfortunately, we're out of time now. Just want to thank our speakers and thank all of you for your participation and your interesting questions. Again, our speakers will address those questions that we couldn't get to on the program offline after the program concludes. Thanks also to our sponsors for making this webcast possible. This program will be available for on-demand viewing through November of 2017. You will receive an email from Pharmaceutical Technology alerting you when this webcast is available for replay. We invite you to forward that announcement to any colleagues who might have missed today's event. Thank you again. See you next time. Goodbye.